This video is going to run you through the evidence of evolution notes. I'm going to run through it pretty fast because you always have that option of pausing the video so that you can write things down. The first thing to do in this chapter is answer the question, what is paleontology? So it is the scientific study of fossils. I don't know how much you guys remember about that old show Friends, but Ross was a paleontologist, and so they were always joking about him loving dinosaurs and such. But this is an ammonite. It is something that used to be alive a long time ago, and we can still find fossils of those around in our area right now. So if this guy is the person who studies this to find out how old it was, or something about the life history of an ammonite, he would be a paleontologist who studies that. What is the geological time scale? This is kind of a picture of what it looks like. It's the paleontologists look at all these fossils and they kind of arrange them in the order that they were created so that you can figure out what the history of life is on Earth. What we know based on the fossil record is that the earlier forms of life were prokaryotic, things like bacteria and archaeans, and life got more complex as it went on. We started to get more complex cells, we got the eukaryotes. From there, life went from being single-celled to multi-celled. Once we were multi-celled, things can start to move from water out onto land. Plants and fungi were the first to move out on the land based on that fossil record, and then animals followed them a little bit after that. In the notes where it says review the time scale on figure 12.2, this is the figure that I'm referring to right here. Some instructors go over this in the first half of the course, some don't. It just kind of depends. You can just Google geologic timescale and find a picture that looks kind of like this, but what I kind of want you to know is there are 3 different eons that life was divided into. The Archean is the oldest, that's when those prokaryotes were in existence. The Proterozoic was a little bit past that one, where you started to get some of the more complex cells. The Phanerozoic is the era that we are still in, and that's where most of the life that you guys would be familiar with is going to be into. The Phanerozoic eon is divided into 3 eras. The Paleozoic is the oldest of those, the Mesozoic is in the middle, and then the Cenozoic is the era that we're still in today. The period is subdivisions of those, and what they're divided by is what was going on during that time period. Anytime there was a mass extinction, that's something that gets us from one period to the next, and if it was a really big mass extinction, it will even take you from one era to the next, which there was a really big mass extinction at the end of the Permian period, and so that's how we also divide the Paleozoic and the Mesozoic, is the mass extinction that happened at the end of that. Same with the Cretaceous. The mass extinction at the end of the Cretaceous period was the one that wiped out the dinosaurs, and that's what got us into the Cenozoic era. Most people have heard of the Jurassic period thanks to Jurassic Park. That movie gets a lot of things wrong, but there were a lot of dinosaurs around, just not necessarily the ones that you guys were thinking about. So just kind of look through here, read through, and figure out what happened with the history of life on Earth based on that geologic timescale. For what are fossils, these are the things that those paleontologists are studying. We've got lots of fossils that are available around in our area. I already mentioned the ammonites earlier. There's a bunch of things like clams that you can find around here. I've found a lot of fossils. I've never found one like the one in this picture. This is a sea spider. It is 160 million years old. But it's a fossil of something that's not alive anymore today. Although there are sea spiders, there aren't any that look quite like that anymore. There's different ways that fossils can form. Compression essentially smushes an organism between a couple of layers of sediment, and then that solidifies into sedimentary rock. So this leaf, it was created that way, and this one actually is special because it still has chlorophyll present in it, and we could see the shape of that chlorophyll within it using an electron microscope. Petrifaction is what creates petrified wood. There's a bunch of that in our area. If you go on a hike, probably you'll find some petrified wood. But this is also what creates a lot of different fossils that are out there. It's just that petrified wood is some of the more common types that you would find around here. Impression, if you go to Dinosaur Valley State Park, which is out in Glenrose, their biggest tourist draw is the dinosaur tracks that are in the river that goes through there, and so those are examples of impression fossils. Cast is basically something got smushed in a layer of mud and then sediment filled in that compression. This is sort of analogous to like if you were to take your kid's hand and smush it into some plaster of Paris and then fill that in with silicone, you could create a mold of your kid's hand. That would be a cast of your child's hand. Intact preservation is fairly rare, but we do have lots of fossils that work this way. Again, going back to that original Jurassic Park, where they were getting dinosaur DNA from mosquitoes that bit the dinosaurs way back when, so that was intact in amber. Otzi the Iceman I talked about a little bit last semester for those of you that had me. He was somebody that was found as a glacier was melting in the Alps, and it turned out he was 5,000 years old, and so we have his whole intact skeleton and body preserved because he was frozen in a glacier for a very long time. Why is the fossil record incomplete? There's really a bunch of reasons, but there's a whole slew of fossils that probably never got made because it takes a lot of very specific circumstances for a fossil to be formed. So a lot of things that die never will be. Like the chance that you become a fossil after you die is actually very, very slim, and so that's one of the bigger reasons why the fossil record is incomplete. There's two different ways that you can determine how old a fossil is. Relative dating is, let's say you find a fossil in this layer in the Grand Canyon. Well, we know how old that layer is, and that means we would know how old that fossil was because it was found in that layer. This is just a different picture that kind of shows you the same thing. So if you found a dinosaur fossil in this one right here, it would mean that it was 360 to 320 million years old, because that's how old that strata is between those two layers. The other way that we can determine it, and usually we use both methods to date a fossil, is using radiocarbon dating or some other radioisotope to determine how old something is. I talk about that in a fair amount of detail in the previous semester, so I'm not going to go into it all that much. Biogeography, for those of you that had me last semester, we did a whole activity where I had you guys draw lines on a map to discover Wallace's line. That's what this picture is alluding to. So the Wallace line, you have Asian fauna, and then you have Australian fauna, and there was a nice little line that kind of separated those. Even though there was an island here and an island here where they had the same kind of climate and plants and things, you found different types of animals in both of those places because of Wallace's line. So that's an example of biogeography. Plate tectonics is what moves the continents around. It's not called continental drift anymore because that makes it sound like they're like floating on the surface of the ocean, and that's not how it works. They are attached to the very bottom off down there, and we have subduction zones and all these sorts of things that move the continents around. The last time all the continents were merged together was about 250-ish million years ago, and it was called Pangaea. Pangaea separated into Laurasia, which is the northern landmass, and Gondwana, which is the southern. Notice which era that's part of. The animals that were starting to evolve during that period, what it meant is they have very specific distributions now because of where the continents were when they first started to evolve versus where they were as they were finishing that process up. So there are a bunch of different types of animals that have what's known as a Laurasian distribution. Think bears. You don't find bears in the southern hemisphere. Like, you're never going to walk around in Africa and run into a grizzly bear. They just don't have them. But we do have them in the northern hemisphere, from polar bears, grizzly bears, sun bears. They all live in the northern hemisphere. Penguins, on the other hand, have a Gondwanan distribution, so you can find them along the southern hemisphere, and that does have to do with when those species were first starting to evolve. Homologous structures are features that are similar due to ancestry, so all vertebrates share a common ancestor. That at least the ones that had 4 limbs, like these guys, they were called tetrapods. And all the tetrapods have the same basic arrangement when it comes to bones. They all have a humerus, a radius and ulna, a bunch of carpals, 5 metacarpals, and then a bunch of phalanges that make the fingers. Even though a cat has very different needs than us, they still have all of those same bones, and it was because both of our common ancestors had the blueprints for how to make all of those structures. Vestigial structures are another piece of evidence that shows evolutionary descent. They are structures that don't have a current purpose, and yet you still have them because from a past standpoint it used to be useful, but now it's not. The example that I'm going to give you for that is the erector pili muscle, which is the muscle that helps make you get goosebumps when you're cold. The purpose of that is to make your hair stand up so it fluffs your coat to make you warmer, but now we don't have enough hair for that to actually make us feel warmer. It just gives us goosebumps instead. So there's not a purpose to that, but it's something that we still have, and it still activates whenever we start to feel cold. So that's what a vestigial structure is. Analogous structures are different from homologous structures. They still look the same, but they look the same just because of similar evolutionary pressures, not because of common ancestry. So both of these moles live underground where it's really dark, so they don't need eyes. So this one doesn't even make an eye. This one still makes an eye, but it doesn't work anymore. They both have feet that are about the same. They're really good for digging, but again, they are very differently structured, but they have kind of the same body plan. And that's because that body plan works really well underground, and so it's useful for things that live underground and burrow around. So they look like that, not because they're related, but because they're both burrowers. How can DNA differences be used to determine relatedness? Well, if you think about it, who on this planet has DNA that is most similar to you? It's your parents. And then your grandparents will be a little bit different, and your great-grandparents will be a little bit different. The farther back you go, the more different the DNA patterns or sequences are going to get between you and your ancestors. If you take that to the extreme, what it means is if you look at two different species, the two species that have the most similar DNA shared a common ancestor more recently. So everybody knows chimps and humans have very similar DNA. That means we had a common ancestor that was fairly recent. On the other hand, if you were to look at human DNA versus the lesser panda DNA, we would have very different DNA. And so we would be on two completely different branches because the DNA gets to be very different. Taken even further, if you look at a human versus a bacterium, crazy different. And so we know that we're very, very distantly related. And so you can just look at the percentage difference in DNA sequences to figure out how related you might be to something else on the planet. How do hiccups relate to evolution? You got the whole story that goes there. So the phrenic nerve comes off towards the top of the spinal cord, and then it goes all the way down and it connects to the diaphragm. That nerve, if it gets irritated, what will happen is it's going to make the diaphragm contract forcefully, and when that happens, it makes a structure up in the throat that's called the epiglottis, which you can't see in this picture. It makes it pull closed. So a hiccup is kind of two things. It's your diaphragm contracting, and then your epiglottis folding over your trachea so that you close your airway off. If this nerve were shorter and it came off towards this part of the spinal cord, the chance of it getting irritated would be much lower, so we wouldn't get hiccups. But the thing is, that phrenic nerve, it's been in our genes since the time when we were amphibians a long, long, long time ago. This is the larva, basically kind of what a tadpole looks like, and they can breathe both in water when they're tadpoles and on land. The phrenic nerve is what helps them gulp water so that it goes over the gills and it closes the epiglottis so that the water doesn't go in their lungs. And for the tadpole, the phrenic nerve is coming off right next to their diaphragm. But because our body plan is different, ours comes off in a different place, but it comes off based on the instructions that we were given way back when amphibians were still the ones that were creating that code. So hiccups are basically something that tells us we're related to tadpoles and frogs because of the way our phrenic nerve is arranged. Here's what's going to happen. There's evidence of